is Leanne Samsa, and I'm an instructor in the biotechnology program at North Carolina State University. Today I'm going to talk about the polymerase chain reaction, also known as PCR. This two-part video lecture will give you a practical introduction to PCR for biotechnology applications. As a reminder, DNA is a double-stranded helix and is normally stored tightly wound in the chromosomes. During replication, the two complementary strands are separated and each becomes a template for a new strand of DNA. The complementary strands are oriented in opposite directions and are conventionally depicted relative to the top strand, with the 5' end on the left and the 3' end on the right. This top strand of DNA is also called the sense strand, the 5' strand, the forward strand, the leading strand, or the forward template. Conversely, the bottom strand is also called the antisense strand, the 3' strand, the reverse strand, the lagging strand, or the reverse template. PCR is essentially an in vitro method for copying DNA, where a specific sequence of DNA is exponentially amplified. The ability to copy DNA by PCR is such an important method that in 1993, Kary Mullis split the Nobel Prize in Chemistry for his invention of the polymerase chain reaction method. PCR is used extensively in molecular biology applications, some of which we'll talk about later. There are 3 steps in a single cycle of PCR. First, double-stranded DNA is separated into single-stranded DNA through the process of denaturation. Next is annealing. Here, short single-stranded DNA oligonucleotides, also called primers, bind or anneal to complementary stretches of DNA. Finally, DNA polymerase recognizes these double-stranded stretches as priming points and adds complementary nucleotides to extend or elongate a new molecule of DNA. So what about the chain reaction part of PCR? Each cycle, the DNA sequence located between 2 primers is replicated. The new products can become template for the next cycle, enabling exponential amplification of the PCR product, at least until reaction components are used up. Under ideal conditions, a single copy of template DNA can be amplified into 1 billion DNA molecules by PCR. There are many applications for this ability to amplify specific sequences from a limited template. To name a few, the PCR product can be used as a source of DNA for cloning or sequencing. It can be used to screen DNA to determine if a specific sequence is present and to verify the size of a PCR product. In forensics, PCR is used for DNA fingerprinting and to investigate it if a sequence is present at a crime scene. It can also be used for site-directed mutagenesis and to quantify gene expression with real-time PCR and reverse transcription PCR. Now let's take a closer look at a typical PCR cycle. First, template double-stranded DNA is denatured into 2 single-stranded template molecules by heating the sample to 95 to 98 degrees Celsius. Then, short single-stranded primers anneal to the single-stranded template to form small stretches of double hybrid and single strands. This happens at between 55 and 58 degrees, depending on the sequence of the primer. Finally, DNA polymerase binds to these double-stranded priming points and catalyzes the addition of complementary nucleotides in the 5' to 3' direction. DNA polymerase activity is optimal at between 68 and 72 degrees, though again this can vary depending on the specific DNA polymerase used. The original template for PCR can be any source of DNA that contains the target sequence. As PCR product accumulates, this new DNA comprises most of the available template. A robust quantity of PCR product can be amplified from as little as 1 copy of the DNA template. But a word of caution about the starting template. More starting material is not always better. Excess template can actually lead to accumulation of undesired PCR products. Let's take a closer look at the denaturation step. There are 2 types of chemical bonds found in double-stranded DNA. Strong covalent bonds that hold the single-stranded DNA backbone together, and weak hydrogen bonds that are formed by the AT and GC interactions between two complementary single-stranded molecules. It is the cumulative strength of these many hydrogen bonds that normally holds complementary DNA sequences together. When DNA is heated, these hydrogen bonds weaken, secondary structures relax, and the double-stranded DNA unzips to form two single-stranded DNA molecules. This is called denaturation, or melting DNA. Denaturation can also refer to the release of protein secondary structures and hydrogen bonds that can form between multiple double-stranded DNA molecules. So whenever we use the term melting, we're referring only to the type of denaturation that is the release of AT and GC bonds to unzip double-stranded DNA. The kinetics of denaturation and melting are actually quite complex and are largely controlled by DNA length and composition. Typically, template DNA denatures at about 95 to 98 degrees. In contrast, primers are designed to melt at about 60 degrees. Importantly, DNA denaturation is reversible simply by lowering the temperature of the reaction. At these lower temperatures, the hydrogen bonds reform and two strands of complementary DNA are joined back together. This process is called annealing. Remember, melting and annealing are opposites. In PCR, we control specificity through primers. Primers are single-stranded oligonucleotides, also known as oligos, that define the sequence that's going to be amplified through regions of complementarity. At about 5 degrees below the melting temperature, which is usually 55 to 58 degrees, primers will anneal to the complementary region on the template, and AT and GC hydrogen bonds will form. This provides a double-stranded DNA priming point that will be recognized at DNA polymerase at the next step. We'll cover primer design in a later video, but just so you know, primer design is essential for specificity of the PCR reaction. The next step in the PCR cycle is elongation. DNA polymerase binds to the double-stranded priming points and catalyzes the addition of complementary DATP, DTTP, DCTP, and DTTP deoxynucleotide triphosphates. Let's call them DNTPs for short. These are added in the 5' to 3' direction. Essentially, DNA polymerase will fill in the gaps and make a new strand of DNA. This new nascent strand of DNA can be used as a template in later PCR cycles. Now that we understand a typical PCR cycle, it's important to note that the first few cycles in the chain reaction are a little bit different. In the very first cycle of PCR, the original template is usually not the amplified PCR cycle, product, and instead a much longer piece of double-stranded DNA. This long template is denatured to form two long single-stranded templates. Primers anneal to their complementary sites in the template strand, and a new strand is formed as complementary nucleotides are added unidirectionally by DNA polymerase. The product produced from this elongation is a hybrid stretch of DNA that contains both single and double-stranded molecules. Fortunately, this represents just a small fraction of the final PCR product, and as the subsequent cycles lead to millions and millions of copies of the stretch of DNA, whose length is determined by the distance between the forward and reverse primers, most of the PCR product is this smaller fraction. Now that we've learned about PCR, let's talk about how to assemble and run a reaction in the laboratory. The reaction mixture contains template DNA, forward and reverse primers at molar excess, DNTPs, DNA polymerase, and the magnesium, salts, or other buffer components that are necessary to support denaturation, annealing, and DNA polymerase activity. The specific reaction components can vary for each specific PCR. The scientists can use different templates along with their own user-defined forward and reverse primers, and there are many commercially available DNA polymerases, each with their own optimal buffer components. When assembling the PCR reaction, always add the DNA polymerase last. After adding all the components to the mixture of the PCR, mix well and centrifuge briefly to collect all the contents at the bottom of the PCR tube, and load the tube into a PCR machine, also called a thermal cycler. PCR cycling conditions will again vary based on the specific DNA polymerase used and your desired PCR product. Always reference the product sheet for your DNA polymerase to verify reaction conditions. Here's a good starting point for a typical PCR reaction. For most modern DNA polymerases, there's an initial activation stage that will activate the DNA polymerase and also helps to denature the original long template. In this case, I've shown this as a 10-minute incubation at 95 degrees. Next comes the 30 to 35 cycles of a polymerase chain reaction. Each cycle has a denaturation, annealing, and extension step. The duration and temperature of each step will vary based on primer sequence, expected product size, and specific enzyme used. The denaturation step is carried out at 95 degrees, and 1 minute is sufficient for complete denaturation. An annealing temperature of 55 degrees is 5 degrees below the melting temperature of an ideal primer. This may need to vary if the primer melting temperature deviates from the 60-degree optimal temp. However, lowering the annealing temperature too far below the melting temp can lead to multiple PCR products, because the primers will be able to anneal to less than perfect complementary sites. Optimal extension temperature can be different for different polymerases, but is usually 60 to 68 degrees, and the optimal extension time depends on the length of the expected PCR product. A good general guideline is to allow 1 minute per kilobase of expected product. Here I've shown a 1 minute extension time for a 1 kilobase expected product. Following 30 to 35 cycles of the PCR, for some applications it's prudent to also include a final extension step, which will allow any products that are in progress to be completely extended. Amplified PCR products can be stored for later use at less than 10 degrees Celsius. A couple of final words about DNA polymerase. Originally, PCR reactions were carried out by moving samples between water baths that were set at appropriate temperatures for denaturation, annealing, and elongation. In the early days of PCR, the DNA polymerases that were available were actually deactivated by the high temperature of denaturation. Most proteins are denatured, also called heat-killed, at high temperatures, but thermophilic bacteria have evolved more stable enzymes that can maintain their function at these higher temperatures. For PCR, this meant that fresh polymerase had to be added at every single cycle. Clearly, this was not efficient. Fortunately, a thermostable DNA polymerase was isolated from a thermophilic bacteria called Thermostable. This enzyme is also called Taq DNA polymerase, Taq polymerase, or just Taq for short. Shown on the screen is the molecular structure of Taq. The thermostable property of this Taq meant it was not destroyed at the denaturation step, which enhances the practicality of using DNA in a molecular biology setting. A couple more fun facts about Taq. Thermostable was discovered at Yellowstone National Park, and it was named Molecule of the Year in 1989. Well, since the discovery of Taq DNA polymerase, many additional DNA polymerases have been identified or engineered to have unique properties for PCR. These properties include fidelity, which refers to the error rate or frequency of incorrect base pairing, exonuclease activity, which is a mechanism of repair, extension rate, which is the temperature-dependent speed at which a polymerase can add nucleotides, the processivity, which refers to the likelihood that a polymerase will detach from DNA during extension, Annealing, in which polymerase will add a single adenosine to the 3' end, and hot start tacks, which have a negligible extension time at low temperatures. When developing a PCR protocol for your own research, in the future you'll need to select a DNA polymerase that's suitable for your application. When it comes time to select this DNA polymerase, please consult product literature to find your best polymerase, and keep in mind that there are many out there that can have these different characteristics. Okay, so if you're interested in learning more, please consider looking at the following 3 resources. PCR Guru is a book available online with a displayed link. Thermo Fisher has a really good short article describing a brief history of PCR. And Paul Rabinow has an excellent book that describes the behind-the-scenes look at the invention of PCR for those who might be interested in the history of this transformative technique. Thank you for viewing. The next video will explore primer design in greater depth.